Hi, today we're going to learn how to do the Punnett squares. So first we're going to deal with a monohybrid cross. And we're going to take a tall pea plant and we're going to cross it with a short pea plant. So the very first thing you do when you read your problems is you need to write down this phenotype-genotype chart. So the phenotype is going to be the physical trait. So the phenotype again is tall and short. And the genotype is big T, big T, big T, little t, and short is little t, little t. And how you determine what the genotypes are is you always take the first letter of the dominant trait and you make that capital. And then if it's lowercase, that means it's the recessive trait. So a tall pea plant can be homozygous or heterozygous. So we're going to do this cross. We're going to take a tall pea plant. So I'm going to place the big T along the sides. And then across the top, I'm going to put the second option, which is little t, little t. And all I do is I take this and move this in, and I take this and move that down. Okay, again, this comes over here, and this comes down. Same thing here. This comes over here, and then this is lowercase. So when you do this particular Punnett square, I get 4 big T, little t, or 4 tall plants. So I have a 100% chance of getting a tall pea plant. However, the F1 generation is all going to be heterozygotes. Okay, well now let's take those two F1s and cross them. So I'm going to do big T, little t, and I'm going to cross it with big T, little t. Okay, so again I bring in the T's. And I'm going to get 1 big T, big T. I'm going to get 2 big T, little t's. And I'm going to get 1 little t, little t. These 3 code for tall. And this one codes for short. So with this, I'm sorry, it's hard to write with my mouse. So this is a 3-to-1 ratio of tall to short. Okay, so let's take another option. We have a heterozygote, and we have it crossed with a recessive. So I'm going to get 2, or 50% chance of being big T, little t, or tall. And 2 little t's, which is short. Okay, that's the monohybrid cross, and you can do any type of problem as long as it only has one trait. And just as a general rule, I always put what's first down the side and what's second across the top. Okay, well, because we do not know the genotype of tall by looking at a tall pea plant, we need to perform a test cross. And a test cross takes the unknown genotype and crosses it with the recessive. So I'm going to take both of my Punnett squares and I'm going to cross it with the recessive, which is short. And then I'm going to put my 2 options that it could be. Tall can be capital T, excuse me, and it also can be, I'm sorry, this should be capital T first, I'm sorry, and lowercase t. So when I do my Punnett squares, I can predict the offspring that I'm going to have. So when I do the actual cross, when I look at the offspring, I can determine what I had by looking at the predictions from the Punnett square. So if my parent was homozygous tall, then all my offspring are going to be tall. However, if I have a heterozygote, I have half of the chance of being tall and half of a chance of being short. So if I get any short offspring, I knew my parent had to be the heterozygote. And again, you always take the recessive genotype and cross it with the unknown, which is these 2. Okay, So now we're in dihybrid crosses. Dihybrid crosses is a cross involving two traits. So for the pea plants, the peas can be round or wrinkled, and yellow and green. And round and yellow are both dominant. So in order to figure out what goes along the sides, you do the FOIL method. So first, then we have outer, we have inner, and then we have last. They're all the same, correct? This is the same thing on the top, except it's all going to be lowercase, lowercase. Now, when you do these, you have to keep the R's together, and you need to keep the Y's together. And each box needs to have 2 R's and 2 Y's. So when you do the Punnett square, you're going to see that the entire 16 squares is full of the heterozygote. Big R, big Y, little y. So instead of writing it 16 times, you can just draw arrows. Or some people just draw one box because it's the same option. Whatever is easy for you is fine with me. Well, let's do the more complicated of the dihybrids. Not hard, but just a little more time that you need to do. So we're going to take 2 heterozygotes. Okay? Again, we have FOIL first, big R, big Y, outer. Oh, this is a typo. This should say Y. I'm sorry about that. Okay. Inner. And then we have last. Okay, and it's the same thing across the top. First, outer, inner, and then the last. Now, I want to point out that on the sides of the square and on top, there's an R and a Y combination. That will give us a double R and a double Y in each box. So when you do that, you keep the R's together and the Y's together. And this is a little time consuming because you have to write all this. Now I want to point out something, and this is something when you do a lot of these problems, you're going to figure out. This diagonal here is always going to be the heterozygote. So I kind of do that first because I don't have to think about it. And that will always be the case if you cross 2 heterozygotes for both traits. Okay? And then I would go back to looking at what I do. And how you complete the chart is up to you. You can go up, down, you can go across. It doesn't matter. Okay, so we're almost done. And one thing to do is you need to make sure your letters, upper and lowercase, look very different, because you can see you can get pretty sloppy in doing this, and then you would be very confused with what you did. And it's always important to put the genotype-phenotype chart up on the problem, because then you can compare and look quickly. Alright, so we're on home stretch here. You can see that I'm getting messier as we go along here. It's not that easy working with a mouse. I don't have a pen. Alright, so that's my 16 squares. Now, the fun part is to figure out what they all code for. So what you do is you need to write down every option that you have. So I took the first box, and that's my option, and I'm going to put a 1 there. And I'm going to cross that off. And then another box, it's different. Put a check mark there, cross that off. Again, still having this. That's one box. Now, remember that this box is a diagonal line, so I'm going to actually do this 4. But I'm not going to write it 4 times. I'm just going to put 4 tally marks, because there's 1, 2, 3, 4. Okay? So now I look. Do I have this one? Yes, I do. So I cross that off, and I just put a tally mark. Do I have this one? No. So now I have to write this one. Give myself my tally mark. Did this one already. Do I have that one? No. So I write that down. Okay. I do have this one. I don't have this one. I hope I don't run out of room here. And I don't have this one. I do have this one. And I do have this one. And I do not have the last one. Okay, so now I want to make sure as I count up my tallies and make sure it adds up to 16. So I have 1, 3, 5, 9, 10, 12, 13, 15, 16. So I have 16 boxes. So I know I've got them all. Now I have to figure out what they code for. So my options, I could be round, and I could be yellow, round could also be green, right? And then my wrinkled, again, really apologize about my sloppy writing. And that is yellow. And it could also be green. Okay, so now I need to look at the chart to figure out what it is. And I look at my phenotype genotype chart. So here, that codes for round and yellow. Mark that off, or I could cross it off like that. So does this one. So does this one. And so does the last one. Okay, this is round and green. Same with this one. Then I have wrinkled yellow. Same thing there. And then my last one is that. So if you notice, I get a 9 to 3 to 3 to 1 ratio. And that is a dihybrid cross. Okay, so now we're going to learn incomplete dominance. Remember, this is the blending. Okay, when they did a red and white, they got a pink. So the heterozygote is going to be the intermediate or the blending color. So we're going to take a red plant and we're going to cross it with a white plant. And you're going to see that all of our boxes are red. Our big R, little r, excuse me, and that codes for pink. Okay? Now let's take and do 2 pink ones and see what we get. So we get 1 red, we get 2 pink, and 1 is white. Okay, and let's do one more thing on this. Let's take a red, I'm sorry, not a red, a pink and a white flower, and let's see what we get. And we're going to get a 50% chance of getting pink or white. Okay, I'm going to start another video for the last 2 types of problems. So go to the part 2, and if you have any questions, let me know.